Hi, I'm Anna Bielen. I'm coming from University of Zagreb, and I will tell you today a few words about the assay that we developed to detect Saprolegnia parasitica in eDNA samples. So Saprolegnia parasitica is a pathogen with a wide range of hosts, important both in aquaculture because it parasites on salmonid fish such as trout, and in environment because it can parasite on free living animals such as crayfish. Currently, the biggest obstacle in the research of this species in the ecosystems is that the identification is based on isolating the pure culture, which is time-consuming and often unsuccessful. Also, there are no specific primers to detect the pathogen directly in environmental DNA. So this was our goal, to detect Saprolegnia parasitica directly in environmental samples. And we have used sensitive technology, digital droplet PCR technology, which also allows absolute quantification of target sequence, and it is quite tolerant to inhibitors in comparison to standard quantitative PCR. So we developed primers that specifically target ITS region of Saprolegnia parasitica, while no amplification was obtained for related species or host species. And at the same time, the assay we have developed had very high sensitivity, meaning that we could detect as low as 15 femtograms of Saprolegnia parasitica genomic DNA per reaction. And we have validated our assay using a variety of environmental DNA samples, like water samples, collected at different locations in Croatia that were positive for Saprolegnia parasitica in more than 50% of cases. Also, we were able to detect Saprolegnia parasitica at the surface of skin and eggs of trout that was healthy and that was diseased. And finally, we were able to detect it on the cuticle of crayfish. This was the first evidence of Saprolegnia parasitica there. And therefore, I can conclude that our assay is applicable in Saprolegnia parasitica monitoring in aquaculture and natural environments, and it presents a starting point for future studies of this pathogen's ecology.